Hello and good morning, everybody. Would you all please stand for the King's Anthem? We would now like to invite our headmaster, Mr. James McDonald, to give his welcoming speech.
Well, it's great to be back up here. It was just a couple weeks ago that we had our first speaker on this series come through and talk to largely the same number of students, and it's fantastic, I think, that at NIST we get to be exposed to this caliber of speaker, to people who have accomplished so much and benefited our world. You know, we're hoping very similar things for our students here, that you'll go out and make the world a better place. So I hope this is very inspiring this morning, and I suspect it's going to be. So a very warm welcome to all our special guests. It was about 10 years ago that this started here at NIST with the International Peace Foundation in partnership, that we have a speaking tour coming through once every 2 years now. And that's been, I think, a fantastic thing for shaping the culture here at NIST and providing that little bit extra to our NIST education. So I'm not going to steal any of the formal introduction, but I did want to offer a very warm welcome to everyone here, our students, our special guests, and everybody else. So thank you very much.
Good morning. My name is Jeff, and we're here for the 5th ASEAN Bridges Dialogue to facilitate and strengthen dialogue between societies in Southeast Asia. I'd like to have a special warm welcome to fellow students from Kissimmee International School, KIS. And today we're here with Dr. Bruce Beutler, a 2011 Nobel Laureate for Medicine. His contributions to science included discovering a family of receptors which allow mammals to sense infection and trigger a powerful inflammatory response. Dr. Bruce Beutler has always loved nature, and he followed in his father's footsteps, studying biology. He went to university at a very young age, and he graduated with his undergraduate degree at 18. He's now the director of the Center for Genetics of Host Defense at University of Texas Southwestern Medical Campus. He graduated from University of California, San Diego, and University of Chicago. I'm pleased to welcome now a pioneer in his field, Dr. Bruce Beutler.
Well, hello. It's a pleasure to be here and to talk to all of you. And I think you've had a string of Nobel laureates coming through here. That's my impression. Each of us has a somewhat different story. I'll tell you a little bit about myself. I'll try to tell you why I love science, and I hope at least some of you do. Or if you don't, then just love something in life and try to do the very best you can at it. I grew up in the United States in a suburb of Los Angeles called Arcadia. And as was just said in the introduction, from a very young age I loved nature. Hard to say why. It may have been family vacations to national parks. It may have been just having a kind of a warm interaction with animals, with pets. I'm not really sure. But at some point, I began to think that it was remarkable that molecules could assemble themselves into living things that seemed much more than the sum of their parts. And that motivated me to study science. When I was maybe 10 or 11, a new science was beginning to dawn. This was called molecular biology. And it seemed to me that by studying molecular biology, I might answer some of the questions I had in mind about the self-assembly of molecules, about what it is that compels inanimate matter to become part of living creatures that appear to breathe, to think, to do all kinds of amazing things as they do. I began to work in my father's laboratory, and I must say that was a great fortune for me to have a father who was interested in science, was really a very distinguished scientist himself. And so in my early teens, I learned how to manipulate molecules that were part of living things, proteins, nucleic acids a bit later on, how to separate them in gels and using special resins. And all of this certainly helped me later on in my career to become so comfortable with working in a biochemistry lab at a young age. When it came time to decide what to do for secondary education, I was very clear about what I had in mind to learn, as I've just said. But how to go about it was a little less clear. Many people thought you should just go to graduate school, learn about biochemistry and molecular biology. Again, it was my father who influenced me on this point and suggested I go to medical school. Now, why that? Well, his reasoning was that if you go to medical school, you're likely to learn broadly about one organism, human beings. You'll learn about physiology, anatomy, about biochemistry further, about pharmacology, and eventually you'll see how the picture all comes together and you'll be able to do anything you want experimentally. And to a large extent that was true. There's one thing I would add, and that is that studying disease, as you have to when you're becoming a physician, is one way of studying nature, kind of an unpleasant aspect of nature. Nonetheless, if you see what goes wrong when someone has a certain disease, then you understand inversely what has to go right, and you do come to understand how an organism works. I went through 4 years of medical school. I would say I liked it in some ways. I did learn a lot, but I decided that clinical medicine wasn't for me, and I decided to do a fellowship in an academic laboratory. I went to the Rockefeller University. There was an interest in the lab I joined in the phenomenon of wasting, weight loss in chronic disease. And I isolated a molecule using an experimental system that we believed might cause wasting. And this molecule has a name now. We called it in those days cachectin. Cachexia is this phenomenon of wasting. But now it's known as tumor necrosis factor, or TNF, which is another of its many activities. This molecule can cause tumor cells to die. Now, my main contribution in that lab was first to isolate that molecule, then to show that it was involved in inflammation, and specifically in a kind of systemic inflammation called septic shock. That got me interested in the whole question of how it is that we recognize infections. When you or I have an infection with a virus or a bacterium or a fungus or some kind of protozoal parasite, we get a fever, we feel awful. If the infection is severe, we can go into shock and actually die. And although these things had been known for a long time, what wasn't known was how our immune system actually detects the infection to begin with. I set out looking with a single molecule called LPS, lipopolysaccharide, to see if I could understand how it was detected. There was good reason to think that that was how gram-negative bacteria make their presence known. And I used a genetic approach to track down the receptor for LPS. This took about 5 years, and I would say that it became an obsession to me. I would dream about what the receptor might be, but the dreams were always wrong, and what really turned out to be right and what worked was to narrow the interval in the genome in which the gene for that receptor might be, and eventually to find it. Persistence is really important. That's maybe the most important lesson, or one of the most important lessons I can give to you. There were a lot of people who told me to diversify or just give up the search at some stage, and I never did. I felt we'd invested too much and I had to track down the critical molecule. So after 5 years we did find it. This receptor turned out to be part of a family of receptors that detect almost any microbe you ever might encounter. And so they're very important. They also are involved in initiating inflammatory diseases and autoimmune diseases of various kinds. And that was the work that led to the Nobel Prize eventually, about 15 years after the fact. Since that time, since we made the discovery and up to the time of the Nobel Prize and afterward, I've been ardent about genetics. I love genetics. I love it because it's a kind of exploration. Some of your teachers may have told you that you need to have a hypothesis when you study things in science. You need to guess about how a biological system works or about how a chemical process works, and you do experiments to test your hypothesis. And that's how progress is made. The beauty of genetics as a tool is that you never need to make guesses. And you instead simply let the organism tell you when something is not right. You create phenotypes. You mutate an organism, such as a mouse in our case. You look for mice with abnormal immunity, and then you find out why. During the last 2 years or so, we've accelerated the process of finding mutations. It took us 5 years, as I mentioned, to track down that very first one. Now it takes only about an hour from the time we see a mouse with abnormal immunity to the time we know the cause. And that probably is my greatest achievement of the last 10 years or so, was making that system work. It's now perfectly possible to march through the mouse genome, which is very similar to the human genome, and find every gene that's important for immune function, however we define it. That's what gets me up every morning. That's why I love to go to work. There may be another surprise, a new gene waiting for me. In fact, there usually is. Once or twice a week, there's something really new and exciting, and it keeps my entire group busy. There are a lot of us now, about 62 people work in my lab today. And I think they all feel very much the same. You never know what's going to happen if you use a random process to try to approach your work and to try to discover all you can about biology. Now, of course, none of this would have worked out for me if I hadn't really loved what I was doing. And I would say that's the first principle all of you should operate from. I often hear people asking, what should I do? There is a quote from Yogi Berra, a baseball player in the United States that many of you probably know, maybe some of you don't. But he once said, when you come to a fork in the road, take it. And that's true. I don't think any of you should be agonizing over what you do with your futures. You should do something. And if it doesn't work out for you, it's not irreversible. You can try something else. That's about all I have to say to you about my own career and about what I would advise off the cuff. And I'm very happy to take any questions you might have. You look like a nice bunch of people. And I'm sure you won't ask me anything too hard. So, thank you. Question here.
Hi, my name is James, and considering the recent outbreak of measles in California, what is your opinion on how to get people to change their opinions on vaccinations?
That's a great question. You know, it's always been that way, that there are people resistant to a certain course of action in public health that experts believe would be useful, and it's very difficult to get them to change their minds. It was true long ago when the very first vaccine was developed by Jenner. There was not long after that, in the 18th and 19th centuries, an anti-vaccine society. There were cartoons at the time drawn of people with cows coming out of their ears and noses and mouths and so on. There's a kind of irrational fear about new medical procedures, only this one isn't really very new. And at the very same time, people are often inquiring, how can I boost my immune system? They recognize that the immune system is important and that it protects us. I would say it's something that even allowed modernity to develop. It's been one of the critical things of our era that we developed vaccines that enable us to live longer and that really enhance the quality of life. So I think it just has to be explained to people in those terms. One has to meet objections with fact, and one won't always win this way, but it's the only way I know. In the back there.
What do you think of like some conspiracies that viruses can be created in the labs, and then like vaccinations and like medicines are created just to make money off of the virus?
Well, there's some truth to that. One of the major motivations in making medicine is unquestionably economic, and some very good medicines don't get made because the economic conditions don't exist to make them. That's a common problem, and it's one that probably has to be addressed at a governmental level. The fact is that it is possible to synthesize viruses these days, and even much more complex organisms than that. If you have the knowledge to make a pathogen, you can make it. If you are really determined to do something with an ill intent, it's very hard to prevent that. And that too is something to grapple with. When you ask about conspiracy theories, maybe you're asking, could it be the case that HIV was synthesized just to do harm to the world? And the answer there is no. I'm sure it was not. The technology was not so good back then to make a virus. And even if you had the technology, you'd have to be more clever than anyone is to make a virus that's really quite that malevolent, that mutates so quickly, that has all of the properties that it does, a long incubation period, ability to cripple the immune system. Those are things that no one could have predicted. So did that happen? No, absolutely it didn't. Could people make dangerous viruses in the future? Yes, probably they could.
Hi, I'm JR. What's your opinion on genetically modified organisms and whether or not they have adverse health effects?
My opinion on genetically modified organisms is that we all are using products of genetically modified organisms all the time. Of course, you probably know the story of maize, which has fed a large fraction of the world. In the time before the conquistadors came to America, the plant was very different than it is today. It was made into this plant that produces these huge carbohydrate-rich kernels, all by human manipulation, all before the dawn of molecular biology and the ability to introduce discrete changes. So humans have done a huge amount with selective breeding In fact, they introduce many more mutations than we can do in the laboratory if they combine wild strains. I don't think, as a rule, it's particularly dangerous to make, let's say, drought-tolerant plants, disease-tolerant plants. Probably if you put your mind to it, you could make something that wouldn't be particularly good for people. But the way GMOs are made now, I find to be quite responsible, and I don't see a great risk in them.
Hi, my name is Peter. Are you worried about the ethical implications of doing animal experimentations with mice, for example?
Am I working on the ethical?
Worried about the ethical implications.
Oh, am I worried about them? Every experiment we do with mice, even from picking a mouse up, handling a mouse in the laboratory, has to be approved by an institutional animal care and use committee, an IACUC. And this is a committee with, I think, a dozen members, some of them from the lay public, some of them university officials, really quite a mixture of people. I'm very sensitive to using mice in the lab. We do things with mice that certainly would not be ethical to do with humans, and yet we take every care to make sure that they don't suffer when we use them. And I don't see any other way to ask the kinds of questions that we are asking, which are so important to humans in the long run.
My name is Charlotte, and my question was, you were talking before a lot about how a lot of the experiments that you do in your lab are on mice. My question is, have you been able to directly apply any of your findings to humans, or is that something that you can see in the foreseeable future?
The answer is yes. There have been some findings that we've applied to humans which grew entirely from work in mice. I'll give you one example. I mentioned this molecule TNF that I isolated many years ago. We realized that it would be important to block TNF activity in humans because it might prevent septic shock, we thought. That didn't turn out to happen. It might also interfere with other kinds of inflammation. To make a long story short, the molecule TNF can be blocked for therapeutic effect in people. It is very effective to block it in treating psoriasis, rheumatoid arthritis, Crohn's disease, ankylosing spondylitis, and a few other diseases. Collectively, that means millions of people. And how is it blocked? It can be blocked with an antibody. It can be blocked with a recombinant inhibitor that we also developed. And that molecule is called Enbrel, or etanercept. All of this had to be tested in mice. We worked first with the mouse receptor, the mouse molecule. We saw what are the consequences of blocking it in mice. Does it affect the well-being of the mouse if you block it? And in some ways it does. It immunocompromises the mice, to be sure. But you have to do things in mice to get to humans. A lot of the mutations we found, and I'm talking about dozens of them, have human counterparts that are important in causing human diseases. And there, too, you have to have the mouse model to move forward efficiently and develop drugs that can target those molecules in humans. Any other questions out there?
My name is Lloyd. Would you eat a lab-grown burger?
A lab-grown burger? I suppose I would if it appeared to me to have been made under good conditions.
Do you think if this product were to be commercialized, would you think that people would have negative opinions about it, like they do about vaccines?
I couldn't quite understand the last part.
So if the lab-grown burger were to be commercialized by, say, McDonald's, would you think that people would be would have negative thoughts on us as they do with vaccines?
Would it have negative effects as to do with?
Vaccines, like
Yeah.
vaccines, people don't.
Vaccines? Oh. I have thought of these things, of tissue culture grown animal cells, as being just basically a kind of animal protein source. And if they're fabricated into meat for people to eat, no, I don't think that they would have any effect on vaccines. Now, I have to say, I can't see, did I?
Vaccines specific that it may do harm to your body. Do you think people have the same opinion about that?
Oh, would they have the same opinions? Would they have the same bias about this new technology for making food? You bet they would. I'm sure they would. I think almost anything that's new encounters some opposition. You know, I remember when I was probably in my teens, maybe a little bit beyond, the first in vitro fertilization was done. The first human baby was produced by Dr. Stephen Steptoe in the UK, and the reaction of some in my family was, this girl, this baby will always be regarded as a freak. Who would want to be born in such a way? And I came from a rather rationalistic family. And now it's very commonplace. I think everybody in the world accepts that in vitro fertilization is, that may not be everyone in the world, but many people accept that it's certainly a way to help people with fertility problems, and it becomes something that's kind of passé. That's the way discoveries go and new technologies go. People think they're a creation of the devil to begin with, and then eventually they get quite comfortable with them. Now, I have to say that one, I doubt it's going to be commercially viable, growing tissue culture cells to eat, but we'll see.
Sorry. A lot of your work was with toll-like receptors, so you've got a really big background in cancer research. I have two questions for you. One is, what increases susceptibility to having cancer for individuals? And the second one is, for those determined to get involved in cancer research, you know, in college or after college, what are some recommendations and tips for us to get involved in cancer research in the future?
Well, you ask what causes cancer, by and large, to begin with. Cancer, by and large, is caused by mutations, and some of these are inherited in your germline, and some of them have occurred de novo as a result of ionizing radiation, viruses, chemicals, other things that are mutagenic. And when genes that control the growth of a cell are mutated, in some cases there are genes that suppress cell division, and if you have both copies of the gene hit, the cell can begin to grow in an unregulated fashion. Usually it's necessary to mutate several different genes in order to get a full-blown cancer. There are other cases where dominant oncogenesis occurs. For example, a virus might insert into your genome, a retrovirus, let's say, or there could be retrotransposition of a piece of DNA that has a strong promoter effect, and it can drive a gene that normally encourages growth. And you only need one such copy of an event like that to occur. And as I said, it can be from a germline problem as well. You may be missing one copy of a growth-controlling gene and you hit the other copy, and this can cause cancer, sometimes of multicentric origin. And there are quite a few famous examples of that. Retinoblastoma is one good example of that. So all of these are causes of cancer. We know of hundreds of oncogenes now. We know only in a very few cases exactly what causes the cancer in terms of the primary driver oncogene and then all the other events that have to happen in order to create the full phenotype of the cancer with all its invasive properties. So there are many open questions in cancer. Something like a third of all people in the developed world die eventually of cancer. And it's very easy to get involved in that kind of research. If I were to give you advice that I took myself, I'd say go to medical school as your entry point. Learn a great deal about pathology and the pathogenesis of cancer, and then choose a lab that's working on what aspect you wish to study and ask if you can join it. See how you like it.
Hi, my name is Sanya. I'm from KIS. I wanted to ask you, what is your opinion on infectious diseases like HIV that spread around the world, not just because of a virus, but also because of social stigma?
Yeah, well, it's not unique to HIV, of course. There are a lot of infectious diseases that carry a social stigma. We could start with leprosy from antique times. Who would like to be labeled a leper? And we could consider all the other venereal diseases that there have been and the social stigma associated with them. I had a professor in medical school who gave a quite memorable lecture about syphilis and said, you know, we could cure it today. We could just cure it today. We could identify everyone who has it. We could give them penicillin and it would be gone, eradicated. And yet that's still never been done. We could still cure it today. Don't know if we'll be able to cure it forever, the way diseases mutate to resistance to antibiotics, but now still true. With HIV, I can only say it's lucky that it had happened in a relatively enlightened time and at a time when there was technology to deal with it. Imagine if it had happened a couple hundred years ago. We would know it was some kind of communicable disease, and we'd be completely helpless to stop it. And it would have burned its way through civilization even much more than it did. So far, drugs have been amazingly effective. It's almost miraculous how quickly we were able to gain control of it, at least if there's access to highly active antiretroviral therapy. Whether a vaccine will be available ever to deliver a definitive answer to it, I don't know. Possibly it will. Certainly it's worth working toward.
Thank you. We hear more and more talk these days about personalized medicine, about designer drugs. And we start to hear from the work of people such as yourself and your lab, that what we previously thought of as one disease is in fact many, many. One of the problems related to this, though, seems to be that the drugs that are being produced are incredibly expensive. Are we at risk of reaching a situation where there will be very, very sophisticated treatments for a very small percentage of the population that can afford these, while a large chunk of the population gets left behind? And have you any thoughts about what we as societies would start to do to address this?
I think there is that risk. I'm not an expert in this area, but at least the last time I kept track of the issue, healthcare in the United States was something like 15% of GDP. And so there isn't a huge amount of room to grow there. We have to decide as a society just how far we want to go in dealing with problems. And the issue you raise about the fracturing of all of the diseases into really small subcategories, each with their own treatment, that could be a big problem, and it could break the bank the way things are done at present. I think, first of all, we have to look for final common pathways. In other words, there might be quite a number of mutations that elevate your cholesterol, and yet if you block HMG-CoA reductase with statins, that's going to take care of the problem always. And there may be a few other final common pathway targets to attack. There's another protein called PCSK9 that's becoming very popular, and people are looking to that. It's a protease that can be blocked by drugs, and it too has the effect of lowering cholesterol when you do that. To me, the great frontier really is on the side of pharma. The ball is in pharma's court. If there could be a miraculous new way to develop drugs quickly, let's say a computational method to assure that a drug doesn't have off-target effects that it's efficacious toward the protein you're targeting. Maybe it blocks protein-protein interactions, which is always a great challenge. Sometime we could imagine, far in the future, we could develop drugs faster and cheaper than we can now, and then there might really be personalized therapy. Another Nobel laureate, Joe Goldstein, talked to me about this a bit, and he asked if I knew the best example of personalized medicine that there is, that's been around for a long time. And I couldn't think what that might be, and he said optometry. And I think that's actually rather insightful of him. Certainly people go and they get their eyes corrected just for them, and there is a technology to do it, and it works. And I see you're wearing glasses, and so do I. And yet we don't have that for drug design yet. That's what's needed. You've identified the problem.
Hi, I'm Ria from KIS. With growing rates of antibiotic resistance, that's growing at an alarming rate, what do you think is the future of antibiotics and antibiotic resistance?
You know, it's a great question. We've had antibiotics for about 70 years out of all the time that human beings have existed. And there's nothing to say they're going to be good for the next 70 years or for the next 200 years or forever. It's a question whether we're going to be clever enough and fast enough to develop new antibiotics that bugs are still sensitive to, whether we can keep ahead of them. And it's absolutely an open question whether that'll be the case or whether 100 years from now people will be back in the lamentable situation they were in before antibiotics. Lucky we're alive now.
My name is Sabir. I wanted to ask, what do you believe is the likelihood of stem cells being able to cure diseases such as Alzheimer's and diabetes in the near future?
In the near future, what's the likelihood of stem cells being able to cure Alzheimer's or diabetes? Is that your question? I think Alzheimer's is an awfully tough target to try to treat with stem cells. Of course, I could be wrong. I've been surprised by all kinds of things in biology in my life, but I don't see exactly how the stem cells are going to replace the neurons and form the same connections and render a brain with the same memories and capabilities as it had before. Diabetes is a very different question. There, in type 1 diabetes anyway, the islet cells have been destroyed by an immune process. And the question is, could you make new islet cells, give them to an individual, cure the diabetes in some way, presumably suppress immunity when you do that? I think that's not nearly as tough a target, and we might see that in the next decade or so, being the common mode of therapy. So it all depends on the target. Of course, we've been working with hematopoietic stem cells for a long time, doing bone marrow transplants for various therapeutic aims, and that works. That's commonplace therapy. Making hematopoietic stem cells from fibroblasts, let's say, from IPS, going the IPS, the inducible pluripotent stem cell route, that's something that still is challenging. But I could see that that barrier could fall too. What are my religious views? It's an interesting question. I'm not a religious person unless you define religion in a certain way. I suppose I could answer that way.
Like, you study like human DNA and stuff, you know, like the smart stuff. I'd like to know, like, especially what you think.
Well, from a young age, I decided that probably there was no deity. That's my personal view, to be very simple about it.
Question about Ebola. Ebola last year, 2014 was the year of Ebola in news. I mean, we heard the most about it. It's been around. What is your opinion on how is how much we don't know about it, but how much we can know about it. Do you think that Ebola is something that will stay around for years, or is it something that you think we can eradicate?
Did everyone hear the question? Okay, then I'll just give the answer. Ebola has been a very well-studied virus. Filoviruses have been studied for quite a long time. A lot of their proteins have been crystallized. There hasn't been a great deal of progress toward making a vaccine that would be usable by the public, but I don't see any huge obstacle there. It would be something very difficult to test, because to have confidence in a vaccine, you need always to do a double-blind controlled prospective trial. And there are people who find that unethical in the setting of a raging infection, a raging epidemic. Nonetheless, it's possible to get around that kind of problem. And will it be eradicated? We've hardly managed to eradicate any infectious diseases, just a couple of them, smallpox and rinderpest. Could it be done with Ebola? Probably. Maybe I could go back to the same question about syphilis. Could we eradicate that? Probably. Will it happen? Not sure. You need to have quite a concerted effort to get rid of it. And there are animal reservoirs in this case. But we could probably create a situation where there are never large epidemics again. Did you have a follow-up question? Okay.
Given the difference between bacterial infections and viral infections, it seems like we've made a lot of progress in bacterial infections because of antibiotics and they can be cured. Viral infections seem to be an entirely different beast. Do you think that there will be, at some point in the near future, the ability to cure some viruses?
Well, there is now. Hepatitis C, you can cure it. It's rather expensive, but you can do it. It's commonly done. There's nothing magical about viruses, and one can hope to cure them. HIV is a really tough case where it integrates into the genome and can reactivate and so forth. But a lot of viruses, certainly I think they could be cured.
Follow-up, what impact do you think the ability to cure a lot of these very impactful viral infections would have on society in terms of population growth and that sort of thing?
Well, I would never rely on infectious disease to be a method of population control. Let's put it that way. So I look to all of these effects on society as being beneficial. Treating disease is what allowed society to develop to the extent that it has. And while we still have all kinds of problems all over the world, believe me, we don't want to go back to the old days where the median lifespan was about 10 years in Great Britain.
What was medical school like for you? Like, what was your medical experience in university?
Oh, what was medical school like? It wasn't exactly what I expected. Here I had grown up and learned quite a lot about biochemistry and manipulating proteins and things like that. And in medical school, suddenly I was called upon to learn anatomy. That was the very first thing. I found I didn't have the best spatial sense, the best memory for what bones are in contact with which muscles and arteries and nerves and so forth. So it was different, and there were a lot of very bright kids in medical school, and I really had to struggle to keep up, although eventually I did very well. But it was a humbling experience at the time. There are a lot of bright people out there, and as you go to a university, if it's a good university, and as you go on to graduate education, you'll find that out too, that you're no longer the very sharpest person on the block there. There are a lot of people who certainly are highly competitive. Now, the big thing about medicine, I can say, was that it reminded me constantly of my own mortality. And some people can deal with that very well. Some people develop mechanisms to go on and go about their work. And I found it quite difficult. I found it a bit painful to be a practicing doctor. I did a year of internship, a year of residency, And then I thought, I have learned enough. And I wanted to go back to the lab, and I thought I could do the greatest good for the greatest number of people that way.
Follow-up question, was it emotionally draining to become a physician?
At times it was, yes, certainly. At times it was, especially for me on pediatric service.
Medicinal would be beneficial. I was wondering what your opinion is on medical cannabis.
Well, I think if one wants to go the route of using any kind of pharmacophore for medical purposes, it should be subjected to the usual rote of tests. One should certainly have a pure compound, not simply say, here we have this pile of marijuana, smoke such and such amount of it. That doesn't pass any kind of standard when you're dealing with real drugs for real medicinal purposes. So it would have to be made according to USP standards, and it would have to be tested. And then one could form a judgment whether it's useful. And if it is, I think that's fine. I don't think the idea of just legalizing it is a particularly sound approach.
I was born with Asperger's syndrome, which is you may know it as a social anxiety disorder. And so far, I actually don't feel very uncomfortable about it. Sometimes I actually wear a label with pride. And however, there may be some certain people who do not actually like having their condition be perceived as a disease, maybe opposed themselves having to be cured. And what is your opinion on this?
Well, nobody should be stigmatized for any disease, is my opinion. You know, now that we sequence genomic DNA, we find that on average about 300 genes are mutated in every one of us. It's kind of shocking how defective we all are. And I look at it that way. Nobody is perfect. People have all kinds of abilities and disabilities, all of us. And as a physician, certainly everyone is taught you don't stigmatize people because of disease.
I wanted to ask about what your personal opinion is about the final goal of medicine. Is it to allow people to live eternally, or is it to allow people to live without suffering in their life and then have a normal, painless death, as they would say it? What do you think would be the ending goal for medicine?
If we said the end goal was to have people live forever, then I think we would be bound to fail. And we would have to admit that we're failing miserably right now. So mitigation of suffering is a very major part of medicine. We accept that we have a certain lifespan. A good doctor will know when it's the appropriate thing to do to treat a certain disease and when it's not, when it's time to give up and to just make someone comfortable. So that's the goal of medicine as it stands. That said, one has to admit that there's not a rule about lifespan the way there is a rule about the speed of light. Probably Brian Schmidt talked to you about that last week. In other words, it is possible that people could live to be 120, 130, 140 years old. Demographers say the average child born today has a life expectancy of about 100 years. That's the average. And we don't know what the limit will be. Personally, I think if it were possible to live in good health, to feel as I do now, or even better, to feel as I did when I was 20, then why not live to be 1,000 years old? Perhaps it should be some kind of a remote goal. And what sets the lifespan of an organism is a very interesting question. We've really changed. What I said a moment ago is true. In Great Britain, in the year 1840, the median lifespan of a human when born was 10 years. Now it's about 80 years. We're talking median, not mean. So we've become a very different species in the last century and a half or so, and we might become rather different in the next century.
Are you currently working on anything?
Am I working on anything? You mean right now?
No, not right now.
Yes, well, in our lab, we use forward genetics, and we use it more than we ever did. And our goal is to understand the immune system as you might any piece of machinery, as you might understand a watch, let's say, with all of its components, these being equivalent to proteins. How do they all work together to create resistance to infection? And we answer that question by mutating mice and by allowing the mice to declare when they have an immune deficiency and then to find the cause. So that keeps me and about 60 other people very busy. Yes, we are working a lot. And we have a lot of individual proteins and genes of great interest to us right now that we're following up.
Hi, my name is Naomi. I go to KIS. My question is, I'm over here.
There you are. Gotcha.
My question is, so there's now rumors about an increase in technology that can detect diseases or illnesses. Do you think that one day technology is going to overpower or take over the places of doctors?
Oh, very possibly. I would argue that it really already has in many respects. And I can see that there's a lot farther that it can go. Of course, the personal touch is very important, and that may not go away for a long time, may never go away. And yet, doctors would be very poor indeed if they didn't have the technology that they do. They would not be able to do nearly what they can do. Think of a simple thing like a CT scan. This is all done computationally. And we used to say in the early days of CT scanning, I was trained as a neurologist, we would say a CT scan is worth a truckload of neurologists. And it sort of was, I have to admit.
My name is Pranav. Exactly how and when did life on Earth originate?
Wow, that's a very tough question. I don't know the answer to that. You know, I can understand very well how we might get a system that would replicate DNA or RNA or some nucleic acid precursor. I cannot understand how a translational apparatus came about. It's a great mystery. Maybe we'll understand it one day by recreating it. Hard to know.
Hello, my name is Aaron. I'm not sure if you're familiar with the technology in the movie Elysium, but they had this machine which could like scan a human and find out the problems wrong with them and immediately solve these problems. Do you think that in the future we would ever be able to accomplish this level of technology?
Don't we do that all the time? We have all kinds of scans.
What I mean is like it just figures out everything that's wrong with a human, whether you have leukemia or a broken leg, and then just solve it on the spot.
Well, will we be able to wave a wand over a person and say what's wrong immediately, instantly? I don't know. Maybe not.
My name is Manit. I'd like to ask you a question of where do you see medicine in the future, in 20 years? Where do you think medicine will be?
I can see room for a lot of progress in a lot of different areas. We were talking about personalized medicine a minute ago. We could imagine a time when cancer chemotherapy will be very much better than it is now. I mean, now the objective is sort of to kill all of the malignant cells, and one winds up killing a lot of rapidly dividing cells that way. But if we could simply target the relevant oncogenes, maybe we would do a lot better. We've actually managed to suppress some kinds of malignancy that way already. The favorite poster drug is Gleevec for that purpose, but there could be a lot more of them to be developed. And so bit by bit we might defeat various causes of infirmity and suffering and really make quite a lot of progress, even in the next 20 years. In the next 100 years or so, it's even hard to imagine how far we might go. But I have to say, it all depends upon maintaining a stable social infrastructure, a peaceful world. If things collapse for one of a number of reasons, then we may never get there.
Thank you very much. Yeah, thank you. It really is remarkable, as I started out talking, and that we have Nobel Prize laureates that come through our school and speak to us. And of course, this is a lifetime of research, but we're 2 for 2, and I know some of the KIS guests, you didn't get to hear the first speaker we had through, but basically what the message is, and if we take sort of your life story as an example, is that if you do what you love and you're passionate about it, great things happen. Don't worry about the outcome. And so regardless of what you're interested in, follow those passions, follow those pursuits. Because I'm pretty sure that you can talk to a lot of Nobel Prize laureates. I'm pretty sure you can talk to a lot of professional athletes, a lot of people that are hugely successful in their fields. And they'll tell you the same thing. Work hard, find out what you're passionate in, and great things happen. And that applies to each and every one of you. Another thing I'd say is that we got to hear your story. And that's a fantastic story because it began when you were a student. And each one of you has a story. Each one of us have a story. And we're writing that every day. And you get to make a choice when you go to schools like KIS and like NIST. You have so much in front of you, so many possibilities because of the options that your parents are giving you, that your teachers provide for you. And so your job, your life's goal is to make the best of that, and following your passion is one thing you get to choose. So thank you again, Dr. Beutler. We really, really enjoyed this conversation. Yeah. Yeah, well done. Yeah.
Folks in the middle on this again, so therefore you need to stand. If you just wait for the guests to come up and join us in the middle here.